And good evening. Hopefully you can hear me. My name is Dave Ritchie. I'm a professor of medicine and haematology based at Royal Melbourne Hospital and Peter MacCallum Cancer Centre here in sunny Melbourne. And I'll be chairing the session tonight. Welcome everybody. Particularly welcome to those who are returning to us for a second time after our kickoff session last week. As you know, this is the second session of a planned 4 sessions focusing on the interests and based on the feedback from patients and their families regarding bone marrow failure syndromes. I was delighted to be able to participate last week and beautifully chaired by Lucy Fox, who joins us again tonight, on sort of general principles and general care issues related to bone marrow failure syndromes. Tonight, we've got an absolutely stellar cast of speakers, and I'll introduce them as we come up to their sessions. And as you will know, we're focusing our attentions tonight on a very interesting, very impactful, and very important form of bone marrow failure called Fanconi anemia. Our experts in the panels will be able to share their experiences and their research and their insights into that condition. And I'm sure you'll find it all very interesting, educational and inspirational. So we've got a number of participants from around the country, interstate, and indeed some international visitors. And it's always a very important point to pause in any proceedings and just thank the people who make it possible for us to meet. And with that in mind, I would like to acknowledge the traditional owners on the land, and in fact the lands on which we meet, and each of us can thank those traditional owners individually as we go through our acknowledgement. I pay my respects, and on behalf of you all, your respects to the elders of those traditional owners, past, present, and emerging. And I would particularly like to welcome any of the Aboriginal elders of other communities who may be joining us today. Thank you for the opportunity to live in such a wonderful place and the opportunity to work here and undertake the research that we're going to share with you tonight. So it's always important to thank our hosts. It's also important to thank our sponsors, by which these educational fora are made possible. With that in mind, our major sponsor is in fact the Victorian State Government, who through a series of grants has made the whole Centre for Research Excellence in Bone Marrow Biology possible, and part of that undertaking is providing educational opportunities to patients, families, consumers, interest groups, researchers, and so on through a series of symposia, and this is one of them. We also would like to specifically thank our platinum sponsor, who is the Royal Melbourne Hospital Foundation, who have been a recurrent sponsor of these events, and they understand the importance of ensuring that patients and their families, consumer groups, and members of the general community get an opportunity to hear about research and to hear about advances in medical practice. So we thank them for their support. And for those of you who joined us nice and early, you will see that we have our ongoing sponsors who have really been excellent partners supporting Maddie's vision in the broadest possible sense, and specifically those being Dexus, Carmen's Kitchen, and Flavour Right. And remember that November is Flavour Month, so keep an eye out for their products, with a fraction of those sales going directly into Maddie's vision and therefore directly to support medical research in bone marrow failure. And we truly are supportive, not just of their financial support, but the true philanthropy and the generosity that they support our activities with, both philosophically, psychologically, as well as financially. So as I mentioned, we have a really stellar lineup between now and about 9 o'clock. We have 3 speakers on our list, and I'll introduce them to you as we get to their sessions. Just a couple of things. Some of you will be brimming with welcome Dr. Addy all the way from Hull in England. First person to put a Q&A in the chat. So that's a good example there. If you just click on the Q&A down the bottom, little icon, a couple of speech bubbles, that's where to pop in questions or comments if you wish. I'll be keeping an eye on those as the chair. We will make use of those questions right at the end of the session in the Q&A session, which you can see there is listed for about 8:30. And so do bear in mind your questions as we go along, pop them in there, and I'll keep a close eye on them and put them in the sort of logical order when we come to our panelists discussion at the end. So hopefully that's all very clear. And thank you very much for joining us tonight. We are, in Maddie's vision, incredibly dedicated to finding better treatments for patients with bone marrow failure syndromes, but also assisting them through their journey wherever we can. And we know it's a long and complex journey. We don't want to assume that we know everything with respect to how patients and their families undertake that journey, and each one is individual, but we really want to learn more about that, because through true partnerships between our donors and supporters, our researchers, and our patients and our families, that we will all find a better way together. So with that in mind, and I think I'm more or less to time with my introductions, really delighted to introduce our first speaker for tonight, who is Amelia Hawkshaw. She's a young woman who lives in Sydney, and as a Melbourneite, I won't hold that against her, because she's done a very generous thing today to come forward and share her story with us. She is somebody who's been diagnosed with Fanconi anemia. As I think a really important point for our discussions tonight, a significant proportion of people are first diagnosed with Fanconi anemia as adults, even though it's an inherited condition. And she's been faced with that diagnosis since her early 20s, and that's a lot to take on for somebody who is emerging as a young adult in the world, and we'll hear more about her in that respect. She's been through a lot, and as she's undertaking her recovery, she's become very generous in her efforts to allow others to learn from her experience, but also to become involved in Fanconi anemia interest groups to raise awareness and bring support to patients and their families who have that diagnosis, and contribute in a number of ways to those educational efforts. And no better example of that than her generosity in presenting tonight and sharing her story with us. So Amelia, over to you, and thank you very much. Feel free to turn on your video. I'm going to turn mine off and make sure I'm mute. Amelia, all yours.
Hello, hi. Thank you so much, David. That was a very warm welcome. I won't hold the Melbourne against you either. Yes, so thanks everyone for having me. And yeah, I'm Amelia. As mentioned, it's hard to know where to start. It's been a little while since I've done this, so bear with me. I thought that I might share my story tonight, a little bit about my road to diagnosis and what my life looked then and now, and with some final thoughts. So as David mentioned, I was diagnosed when I was 23 as an adult, and that does explain some things earlier on in my life. So a little bit of the timeline for me was the way I presented and the way I came to my diagnosis was a bit of a hard road. In late 2015, I started getting like stomach pains and things like that. And I thought it was a diet related thing. But over time, we worked out it wasn't diet, and I had a CT scan and was diagnosed with bowel cancer at the beginning of 2016. Following that, I had surgery to remove the tumor, and while recovering from the surgery, I had a complication and had a second surgery in the same place due to an abscess that burst. And then I was due to receive 6 months of chemotherapy, And if you know anything about Fanconi anemia, which I might call FA because that's just a little bit quicker and simpler. Yeah, if you know anything about FA, you know that we do not take well to chemotherapy. We are cytotoxic. For those who don't know, that means that it affects us really badly. Chemotherapy affects us quite badly. And 2 days into my first cycle of chemo, my bone marrow shut down, and they didn't know why. It just crashed. Yep, so we stopped the chemo, and I went through almost 2 months of being very sick for a variety of different reasons. Yeah, from hospital pneumonia to my lungs filling up with fluid to different infections and all those kind of strange things, allergic reactions, etc. All because my bone marrow had left my immune system very weak. Yeah, after that, I managed to recover strangely, which was quite a relief since we didn't really know whether that was going to happen. And it was a couple of months after that, in November 2016, that I was diagnosed with Fanconi anemia. So yes, it was a little bit of a hard year. It was quite a shock, being quite young for bowel cancer to hit. At the time when I was diagnosed, I had just finished my bachelor degree. I'd graduated, I had my first job in the industry, I just started working full time, and then suddenly cancer hits. And cancer is a big word for everyone, let alone for someone when they're 23 and haven't really been around much illness before. So yeah, it was quite daunting. Yeah, so in terms of that year, there was a few hard things going on. I felt isolated because of the cancer, with most of my friends not knowing really what to do. I was quite physically unwell and didn't have a lot of mental capacity to speak to people because I was just trying to survive, basically. I had to consider and face my mortality head on. Also very scary for a 23-year-old. And of course, we had a lot of uncertainty before the Fanconi anemia diagnosis, which is quite scary. Throughout that time, some things that helped me were my family, my support networks, my boyfriend, now my husband, my church community, friends. I had a psychologist, and yeah. So those were some of the things that supported me during that time. So yes, so recovery from bone marrow failure, and then the diagnosis of Fanconi anemia, which for some would be really scary, for me actually brought quite a bit of relief. As you can imagine, the not knowing was what I really struggled with. Yeah, so the early days post Fanconi anemia diagnosis, I suppose, in 2017 and 2018 was a lot of adjustment. As we know, there are a lot of different things that could go wrong with Fanconi anemia, and so there was a long process of trying to find my exact genetic pathway and seeing whether that could tell us a little bit about what I might expect or what kind of checkups I should be getting and things like that. Yeah, but the early days were a lot of research, especially from my parents, to work out the best way to support me. Yeah, so throughout the time when we were adjusting, we were meeting lots of new doctors and getting lots of different doctors on board to perform different checkups, which I can go through a little bit later. And one of the key turning points was getting linked in with the online communities for Fanconi anemia. So there's two crucial things that happened. One, I got connected to the Fanconi Anemia Research Fund. They are an organization in America who support Fanconi anemia research. and FA patients as well, with resources and advice, linking them to doctors, linking them to research projects as well. And most crucially, the meetings that we have. There is a scientific symposium for clinicians. I'm sure some of you already know these things, but I'll repeat them for those that don't. And a family meeting and an adult meeting for adult patients. That is the one that I went to at the end of 2017. And I went from having met one person with a Fanconi anemia diagnosis in Sydney to over 30 people who are adults going through what I was going through. And it just changed the way I saw my condition and the way I saw adjusting to life with this condition. And it made it much less isolating, even though everyone was spread across the world. Yeah, it was a real crucial turning point. The second crucial turning point was learning that we had a community in Australia when I went to the St. Vincent's Institute meeting in Melbourne, and met not 30 adults with Fanconi anemia, but a fair few people. And yeah, I'll get more into what we set up as part of that a bit later. So yeah, 2016, 2017, and 2018, getting into a rhythm, adjusting. And then I was thrown another curveball in 2019. So just as we'd gotten used to this rhythm of learning about how to manage lifestyle factors and monitor with checkups, I had to have brain surgery, some hydrocephalus that we do believe was explained by the Fanconi anemia, was just not being compensated for in my head anymore. So I had surgery in 2019 for that. And I believe it was around that time, though I may not have the dates quite right, that we set up Fanconi Anemia Support Australasia, which is an Australian and New Zealand version of the Research Fund in America. Or we try to do some similar things and fill in some gaps for Australian and New Zealand FA patients. Yes, so we similarly continue to run that meeting in Melbourne every second year or so, and have resources and tailored help specific to Australia and New Zealand. Cool. So that's 2019. Since then, I've also had 2 skin cancers. One was a BCC, gone straight away, but one was a melanoma. So I had a surgery for that. Timeline. In terms of the timeline, in 2020, I got married and have started that wonderful adventure with my husband, Zach. And there was another readjustment there as part of my story to having more responsibility for my monitoring, which we're still adjusting to. So to clarify my monitoring and my checkups and what that looks like, I see a variety of doctors across the year. Some of them I see every 3 or 4 months, some every 6, and some every 12 months. They range from a hematologist to an ear, nose, and throat doctor, to a dentist, to a gynecologist, to my bowel, my gastroenterological surgeon, dermatology. I have 2. My geneticist, who also does my breast cancer checkups. I also have an endocrinologist and a brain surgeon that I follow up with. So they're just, they're the main doctors that I see, and we have a few other ones kind of in the back of our heads if we need them. So what does life look like now for me? So just pull up my notes down here. Ooh, one sec. Cool. Yeah. So what does life look like now? In terms of Fanconi anemia, it looks like continuing to monitor for all of these things that could go wrong. It looks like uncertainty because I'm a little bit of an unusual case, so it's hard to monitor when we don't know if I'll be quite severe in my symptoms or not so severe. So a lot of uncertainty there, fear of the future, but it also looks like planning for the future. We're currently working on working towards starting a family at the moment, which is complicated as I have fertility issues also due to Fanconi anemia, but that's a big thing for us. And life also looks like working. I work 3 days a week as a youth worker in Sydney, which for me involves mostly going into schools and running programs and things and supporting young people one-on-one, which I'm doing at the moment. Cool. So in terms of how these experiences changed me, both the cancer experience and having FA and all of the things that come with that, there are a few key areas that I wanted to touch on. It changed the trajectory of my career. So I was going into social research before I got sick, and that's due to my experiences partly with Canteen, the cancer charity for young people, and my experiences there, and partly through my faith and youth group influence. I realized I wanted to work frontline in community services. I wanted to help people and I wanted to work with young people. So it's changed my career in that sense. It's changed my life in terms of my faith. I did have a faith before I was diagnosed with cancer, but I really struggled with it during 2016 when so many hard things were happening. During that time, I was supported by many people in my church community. And while I did struggle with things, I came out the other end with a stronger relationship with Jesus and with my community. Thirdly, another thing that changed in what my life looks like is advocacy work. I am really committed to telling people about Fanconi anemia and supporting people with Fanconi anemia. Yeah, and I hope to do that a long time into the future. I think David mentioned these a little bit, but I was in the Canteen Youth Leadership team. I was on the Fanconi Anemia Research Fund Adult Council for a while. I'm in the committee for Fanconi Anemia Support Australasia, the Australian charity. I'm involved with the online community, and I've done some long case interviews with medical students and things like that. And I tell every doctor and nurse that I meet about Fanconi anemia, because the more people, the better. One of the best things that I love when I do go in and go to my checkups or meet a new nurse or anything is when I mention that I have Fanconi anemia and they say, oh, I've heard of that. Like, I know what that is. And like, tell me more about it kind of thing. It's so encouraging when, yeah, someone wants to hear your story. And secondly, yeah, when they've actually heard of something so rare. So that's one for the clinicians. Yeah, so in terms of how it's changed me also, it's solidified my priorities in life and helped me work out what I wanted to focus on and value, being my family and my faith and helping others in practical ways. In terms of new patients and what helped me in the beginning and things like that, my notes get a little bit choppy to work all of that out. For me, a big thing was getting myself educated. Some people, that's not the right way to go, and they'd rather just live their lives without knowing. But for me personally, I did everything I could to learn about what was happening to me, and my family helped me prepare for all of those possible things with the monitoring and the changing lifestyle factors and things like that. yeah, so that helped me feel like I had a sense of control over what was going on. So for clinicians, it might be helpful to just have those resources at the ready, whether that's connecting them or just sending them a simple link to the Fanconi Anemia Research Fund website, or if they're in Australia, the FASA, Support Australasia charity. Yeah, or simply just having a flyer there, things like that can help. More into the nitty-gritty clinically, in terms of, I was kind of asking myself, like, what would have helped me in the beginning, especially as a young person. I didn't have a lot of resilience at the start to what I was going through, because there were things that frustrated me that I just didn't expect to be frustrated by. For example, there were just things that I didn't know that I didn't know. Like the first time that I had a cannula put in my arm was the night before my first surgery. And I was like, what is a cannula? Like, this has to sit in my arm overnight. Can I move my arm? I don't know. So all of that was really daunting. So I guess it's about working out how to prompt your patient to see what information they might want, even if they don't know what questions to ask. Yeah. Emotionally, in terms of the early days, I always recommend connecting to the FA community as fast as possible. And I do wish I'd been connected in with them earlier. So yeah, that goes back to connecting your patients in. For anyone out there who is a new FA patient or a parent of a child who's just been diagnosed, I definitely recommend you reach out to the Fanconi Anemia Support Australasia website, or send us an email, send me a Facebook message. We'd love to get you connected. I know that FA can be really isolating in a lot of ways, from having to educate your doctors because they haven't heard of it before, to your friends or family members, extended family, just not understanding what's going on or how they can help. All of that can be exhausting and overwhelming. But there is a community of people out there that you can connect with who know what it feels like to be overwhelmed by doctor's appointments, or having to advocate for, yes, let's get that mole cut out and checked rather than let's wait and see. Yeah, or just understanding how the possibilities of what could happen to us might affect our relationships with other people. Yeah, there are people out there who can share in that grief with you. What am I missing? I think I got most of my points out. Sorry, I was a little bit all over the place there. As I said, Fanconi anemia is a bit of a crazy time, and it affects, I guess the other thing to remember is that every Fanconi anemia patient is different. We all have different genetic pathways and it manifests in so many different ways. Yeah, so everyone's different. Everyone has a unique experience of it. Yeah, so please listen to your patients and make them heard. And definitely shop around for those people that will hear you properly. Cool. Thank you.
Well, thanks so much, Amelia. We really appreciate all of those insights, and you've touched on so many critically important elements of Fanconi anemia, in fact, bone marrow failure syndromes generally. We are going to hold questions for the Q&A session at the end, but I can already see them coming in, and I've got a bunch that I'd like to ask you as well. Do have a look at the Q&A section if you can, for the participants, because there are some really helpful links being posted there for people to have a look at, to get some additional information, and perhaps even join up to the Fanconi Anemia Support Australasia group. Excellent community, and one that does a huge amount of great work. So we really appreciated Amelia's time. I really would like to emphasize that point that Fanconi anemia patients are so different. They present in different ways, and that's one of the reasons why we hold these symposia, to get the information out and what to look for, what to be aware of, what the warning signs might be, and for people to look a little deeper and to help make those diagnoses, that people get accurate diagnoses as early as possible and linked into the support and medical groups that they need. So we are going to just change gears slightly, not off the topic, of course, because this is Fanconi anemia specific session tonight. But it is my great pleasure to introduce someone who's been a pillar of early research activity in MRV. Of course, his research activity extends well beyond there was such a thing as MRV. And Associate Professor Andrew Deans is who I'm talking about. And in fact, a fellow in his lab who's gone on to great things, was one of the very first recipients of grant research funding from MRV in the very earliest days. I won't mention names because we're focusing on Andrew. So Andrew, his story goes back a long way with Melbourne and research. Initially some cancer research with Grant MacArthur at the Penn McCallum Cancer Centre, and then really moved on to the bone marrow failure, or at least gene repair issue, when he went to London as a postdoctoral researcher. Very fortunate for all of us and Australia generally. He relocated back to Australia and started the genome stability unit at St. Vincent's Hospital. And you've already heard St. Vincent's referred to by Amelia, and they've really been a mover and shaker in the Fanconi anemia world, really because of a lot of Andrew's insights and efforts. He's got a big research group at St. Vincent's Institute, a dozen or 15 or so people working on different aspects of gene stability and repair mechanisms. And one of the great opportunities that have come to light in the last few years in bone marrow failure syndromes has been the ability to identify specific gene abnormalities that might be amenable to gene editing, fixing the genes that don't work properly. And that's exactly what Andrew's going to talk about tonight for us, because it really is a very exciting area for bone marrow failure syndromes generally, and more specifically in Fanconi anemia. So with that, we thank Andrew for his time and for his insights. Thank you, Andrew.
Thanks so much, David. And thanks again to MRV. This is a bit of a follow-up talk from last week, so I'm hoping that the majority of the audience here tonight saw my presentation from last week, but I am going to recap a little bit. I just based on what David mentioned earlier, I acknowledge the Gunner-Kurnai people as the traditional owners of the Gippsland area of Victoria from where I'm presenting from tonight. And I'd also really like to acknowledge Amelia for going in first and sharing such an inspirational story and giving a lot of insight that we don't often hear as researchers. Okay, so as David mentioned, I wanted to start by acknowledging my team, who's done a lot of the work that I'm going to be talking about today. My lab split into two teams. And of course, there's also Wayne Chrismani's lab. It's also at St. Vincent's Institute working on Fanconi anemia. And the two teams work on related areas, understanding the basic biology of DNA repair and development of therapeutic gene editing. And of course, the intersection of these two areas of research is the topic of interest tonight, which is Fanconi anemia. As you're probably all aware, Fanconi anemia is the most common of the inherited bone marrow failure syndromes, but it's also still a really rare disease, and it affects only about 1 in 80,000 births in Western populations. Some of the characteristic features of FA are short stature, developmental defects such as extra or missing digits, skin spots. But of course, the one I'm going to talk about, focus on tonight is bone marrow failure. The median hematological onset is just 8 years of age, and this leads to progressive bone marrow failure over the next 10 to 15 years. And the current treatment for this is bone marrow transplant that we talked a lot about last week. It's expensive and requires a lot of hospital stay and is also quite risky. So like Amelia hinted at, Fanconi anemia cells are very sensitive, or patients are very sensitive to chemotherapies. And that's because chemotherapies work by damaging DNA. And the Fanconi anemia DNA process or pathway is involved in protecting cells from DNA damage. And this can be DNA damage caused for environmental things such as DNA cross-linking drugs, which are used in chemotherapy, but also aldehydes, which are byproducts of metabolism just from the food that we eat. So they're kind of impossible to escape. But these phenotypes can be seen clearly when we look at cells from Fanconi patients down a microscope. This is what a normal DNA chromosome looks like, but this is what chromosomes look like in Fanconi anemia patients when they're treated with these DNA damaging agents. They form these really unusual DNA structures. And because these are not repaired properly, especially in stem cells, it leads to the death of those cells, which is why we get all of these different phenotypes. But it was realized many years ago that a subset of Fanconi anemia patients called mosaics have actually a much better outcome. So they have a later onset of hematological problems. They also, they're much less likely to require a bone marrow transplant. So this graph here in the red line shows the age at which a whole host of Spanish patients required a bone marrow transplant, Fanconi anemia patients. But the line in green are patients who are mosaics, and they form this green line. They're much less likely to need a bone marrow transplant, and they also live longer. So what does it mean to be a mosaic? Mosaics can be thought of as actually naturally gene-edited patients. Somewhere along the line during the development, a rare genetic mutation, second genetic mutation occurs in one cell quite early, perhaps on in development, perhaps in blood cells. And this leads to restoration of a normal gene function and normal protection from these DNA damaging agents. And what happens is if you start with one cell, but because this cell has a natural survival advantage over all of these other stem cells, which are normally likely to fail over time, they eventually outgrow the mutant cells and repopulate the blood. So this process, if you're familiar with the concept of evolution by natural selection, it's exactly what's going on here. And another way, an analogy, which as you heard from last week, I like my analogies. You could think of this a bit like black rabbits in the snow. Black rabbits quickly wiped out by foxes because they are easy to pick out. But if a rabbit is born with a mutation that makes the rabbit white, those rabbits are now able to survive and go on and thrive. And that's kind of what's happening here. Mutations allowing these cells to go on and thrive. And really only one cell is needed for this benefit to be seen. I'll take you back to this tree that I introduced last week because I'm going to talk a little bit about stem cells again. Human embryonic stem cells are what we start out as. And then we go on as an embryo develops, all of these other stem cells are derived from those original embryonic stem cells. And the cells that we're interested in are these hematopoietic stem cells. And in Fanconi anemia, it's the failure of these hematopoietic stem cells to survive that leads to the bone marrow failure. And it's really an intrinsic problem of these cells. And if we look at the number of stem cells that a non-Fanconi patient is born with, there's enough of these stem cells to provide all of these different blood cells across their lives. But in Fanconi anemia, they're born with enough stem cells to provide blood at birth or early on in life, but the stem cells slowly deplete over time towards zero, and this is when bone marrow failure occurs. And this would be the point at which the child would be diagnosed with a full-blown bone marrow failure. Again, as I mentioned last week, the capacity to continue making hematopoietic stem cells throughout life is controlled in our DNA. And I'll go, again, just quickly back to my analogy about thinking of DNA literally like an instruction manual for generating this complex tree of life, where one gene provides one instruction to a cell. A chromosome is a set of these instructions, and in humans we have 23 of them, and a genome is the entire set of these instructions that's passed, half of which we receive from our mum and half from our dad. So every cell, including every stem cell, has a full copy of the genome that encodes all of these genes that we need to survive, and makes us a unique person, makes us unique, who we are, what we look like, how we various features, and how we function. There's a whole series of genes. There's 22 different genes which have been identified to predispose to Fanconi anemia. And importantly, it's when these genes are not functioning in an individual, that's when Fanconi anemia occurs. So everybody inherits a FANC-A gene and everybody inherits a FANC-B gene, but it's when you have a mutation in the gene in both copies, one that comes from your mum and one that comes from your dad, that you end up with Fanconi anemia. These genes, unlike other bone marrow failure syndrome genes, are all involved in a very specific process of DNA repair, which I'm not going to touch on very much tonight, if actually at all, but I'm happy to. I've talked about that in the past. So again, to show this literally, here's some sequence from the FANC-A gene. It's made up of this 4-letter code, but provides this specific instruction for making the FANC-A protein, which is a DNA repair protein. In this version of the FANC-A gene, there's a mutation in the instruction. And last week, I explained the concept of genes like cake recipes, where a normal gene is, oh, I've lost my recipe. Okay. In a normal gene, the recipe would be entirely readable, but in the mutant version of the gene, you wouldn't, you're not able to read the recipe properly. Now, in gene therapy, which I also talked about last week, gene therapy is an approach whereby mutations in genes are not corrected. However, an additional copy of the gene is inserted into the patient's cells using a virus delivery system. So stem cells are taken out of a patient. They are modified with the virus in culture, in a laboratory setting. They're grown up a bit so that they have a few more than what they took out, and then they're put back into the patient. And in this successful gene therapy trial that was conducted in Spain, 4 patients in this published study, I think it's now up to 8 or 9 patients that have been successfully transfused with gene therapy containing cells. You can see that although a very small number of cells are put back into the patient, if we follow that patient over a couple of years, the gene edited cells are what restores the blood. And while the uncorrected or the existing patient cells continue to go down in number towards that 0 or very low number of bone marrow failure, the edited cells have this great evolutionary advantage, survival advantage over those unedited cells, and they can grow from quite a small number to a very large number quite quickly and stop bone marrow failure from occurring. And so if we go back to our graph, if we perform gene therapy prior to bone marrow failure, we can restore the normal blood production and hopefully prevent bone marrow failure for the entire lifetime of that patient. So it would be a permanent therapy. So with gene editing, here's my recipe back again. Instead of using a virus, we actually use a non-viral method to get inside the cells. And we're going to completely now fix this mistake that's in the recipe. We're going to go in, we're recognizing that there's an R missing here. We're going to write it back into that recipe. That will shift the frame back into something that is readable, and we'll be able to make our nice Victoria sponge. This is a programmable process, so every patient has different set of mutations because of this really strange property of Fanconi anemia that can be caused by so many different genes. So we need to be able to target all of these different genetic mutations using this approach. And there we go, we've fixed up the recipe. So what would a good gene editing tool look like? What would, I've talked about in using an analogy, but now I want to talk about the real scientific problem that we have, because it's not just like inserting a letter into a book with a pen. That, you know, we can do that. I could pick up a pen now and do that and correct I actually spend a lot of my time correcting other people's work. So I'm quite familiar with it, and everyone can do it, and it's very straightforward. But doing this inside cells and doing it in a difficult population of cells, such as hemopoietic stem cells, it's a big challenge. And, it's why this type of therapy and research into this area is taking, it takes many years. So first of all, we need a gene editing tool to be efficient. We need to get editing in as many cells as possible, because that will just give a much greater likelihood of success. It needs to be easy to use, and that includes both a low cost compared to bone marrow transplant. It needs to be scalable so that we can apply it to as many patients as possible. But most of all, it needs to, in reality, have a simple concept so that it can be understood and how it works, so that we don't have any risks associated with it. That comes down to the point at the end about it being safe, that we get editing only at the desired location. And to come back to the fact that we want to be able to edit lots of different possible mutations, it needs to be programmable, which means it needs to not just correct a single mutation, but many different mutations. So many of you may have heard about this technology called CRISPR-Cas9. And this is actually a bacterial protein that's normally involved in resistance of bacteria to virus infection. So it seems a bit of a strange tool to be using for fixing bone marrow failure syndromes. But the beauty of this protein, and it recently, the discoverers recently were awarded the Nobel Prize for their discovery, is that this represents the first RNA programmable enzyme that can be targeted to a very specific DNA sequence in the genome. So, for example, if I wanted to edit this word occurs in this entire paragraph, I can place my cursor here and I can start editing. The CRISPR enzyme does that. It finds this word amongst the whole series of instructions that are present in this manual of life called the genome. So CRISPR-Cas9, when it's applied to human gene editing, is very efficient, and it can induce editing in very many cells. It's incredibly easy to use, and so it's been a fantastic research tool to researchers, probably one of the greatest tools for molecular biologists and our understanding of human development that we have ever seen. It is programmable in that it can be used to find regions of the genome. However, it can only create insertions or complex reconstructions, which means that it's only usable in a very small fraction of cases. And unfortunately, a series of or many experiments over the years have shown that although it can be safe in some settings, for the majority of sequences that it targets, there are also so-called off-target sequences that induce double-strand breaks at the wrong place in the genome. So occasionally it recognizes the wrong word and makes a break there, which could have cancer-causing consequences. But today I'm going to talk to you about work that we've been doing in the lab with a newer technology, and we're calling this gene editing version 2.0. But it goes by a specific name of prime editing. It also uses Cas9. However, this version of the protein has been modified so that it can't induce double-stranded DNA breaks. It only induces single nicks into DNA. And it now includes a template writing mechanism called a reverse transcriptase. And this allows almost any mutation to be rewritten back to a normal sequence. So I'm ticking all the boxes here. There's probably some others that I'm missing that maybe it's not perfect for, but I think for the purposes of what you're going to see later on, I hope you'll be excited that this really could be the real deal in terms of gene editing. So explaining how prime editing works, I'm going to get quite technical, and some of you are probably saying, well, you've already been really technical. I'm going to get more technical because I know there's some scientists in the audience who already have an understanding of how CRISPR works, and I'll try to just go through this quickly. But for the non-scientists, I hope you get a flavour of really how clever this technology is. So firstly, it uses a Cas9 fusion protein bound to a guide RNA. And this is the guide RNA that is the bit that locates the correct sequence in the genome that's to be edited. However, attached to this guide RNA is an extension that actually includes an edit template. And this is the bit that's used to correct the sequence that needs to be introduced to replace the mutation that's causing the genetic disorder of Fanconi anemia. But also present in this fusion is the reverse transcriptase enzyme, and this is the enzyme writer that copies the RNA into the genome. The enzyme is added to cells, and I just again point out that it's a version of Cas9 which only creates a nick, not a double-strand break like a normal Cas9 protein. And this leads to the creation of a flap, or like a bit of single-stranded DNA that opens up, which can anneal to the primer sequence. And the reverse transcriptase then copies this sequence to create into the genome. And this is the writing part. So the first bit was reading by the enzyme, and now it's writing. And then this can be incorporated into the genome, and there's a lot of stuff that goes on in between for that. So we wanted to come up with a really simple way to be able to determine the number of cells that are edited given in a given experiment, using a given experimental approach. And this is because there are actually hundreds of parameters that need to be tested to find the best editing approach for a given cell type, and in our case, hematopoietic stem cells. So Astrid Glasser in my lab turned to a really special gene that a lot of researchers have used over the last few decades. In fact, another Nobel Prize winning discovery. And this protein comes from jellyfish, and it encodes the instructions or the recipe for making a fluorescent protein called GFP because it fluoresces green. And this is encoded by a DNA sequence like any other gene. And Astrid discovered that you could actually change 2 letters in this sequence, and the protein now became blue fluorescent protein, or BFP. And the DNA sequence of this instruction differs by only 2 letters. And of course, these are the 2 letters that we can target using this prime editing approach. And if you put the GFP gene into human cells, you can see that they glow green under a certain light, because this recipe is being transferred into the human cells and being followed by the cell machinery. And what you're looking at here is just normal light microscopy, and you can see all the individual cells growing on the dish. And then if we look at it under a special light, we can see the GFP is present in all of those cells. But then she performed editing on these experiments using the prime editing technology, and you can see that a percentage of the cells turned from being green to being blue. And if we look at them both, you can see that the green and the blue is mutually exclusive. Another way that we can look at these results is using a method called flow cytometry. And this is where, instead of looking down a microscope, each of the cells is analysed one by one as it passes through a laser. And what we're looking at here are cells which are, well, we can look at the green or the blue fluorescence. On this axis, the cells are wholly green. On this axis, the cells would be entirely blue. This is what it looks like for these unedited green cells. They're all falling up here in the green channel. But remarkably, using this prime editing approach, we can actually convert over 90% of the cells from a single experiment from 100% green to 90% blue. But next we really want to understand whether this approach is going to work in hematopoietic stem cells, because we can get this technology to work in kind of immortal cells quite easily. So Astrid in the lab worked with Jörg Heyerhorst and Louise Purton, who are also both at Sir Vincent's Institute, to make an EGFP mouse. And this is a cartoon, but this is actually what these mice look like when they're young. And for those of you not familiar with research, we do a lot of research into human biology by trying to understand what happens in mice. This is what the mice look like when they're young. These are the ones that express the GFP. These are the ones that don't. And we can isolate this hematopoietic stem cells from these mice using this flow cytometry technique with some different markers that specifically isolate the stem cells. And what we can see is when we do that, that these cells are uniformly green as well. So what we have are some kind of cute green mice. They've got green blood and they've got green hematopoietic stem cells. So Lulu is a PhD student in the lab, and she's been working extensively with primary stem cells from these mice. And I wanted to share with you tonight some really exciting new results that we haven't really shared at all with anybody else yet. And so in these experiments, Lou's really been able to go from getting a low level of editing very early on when we started this work about a year ago, And as time has passed, we've improved and improved. And we've got to the point now that we're able to get more than a third of the cells edited from green to blue. And we're just at the point that we're going to put these hematopoietic stem cells back into mice, just to confirm that after this editing approach, they're still able to form functional blood in the mice. And this is a really exciting project that Lou's been focused on for the last couple of years. But of course, you're probably saying, well, why do you want to muck around with green proteins and making blue ones? Like, that just sounds like you're going to, it's Spider-Man is next. But the reason we started off with that was because it's as you can see, we can do really quick and simple experiments looking for the conversion of green to blue cells, and we can understand the best way to perform editing in hematopoietic cells before we then move on to trying to do it in Fanconi anemia cells. But another problem we had was that there's actually no mouse model that is susceptible for gene editing available. And that's because all of the existing Fanconi mouse models have a complete loss of the gene in question. They don't have something that can be corrected like is what's found in most patients. So we've also made a second mouse, which we used a CRISPR-based strategy, which probably don't have to understand, but we introduced a mutation into mice that's also found in humans, in a human Fanconi anemia patient. And we created a 3 base pair in-frame deletion, which led to the loss of the instruction, a normal instruction for making Fankel protein. Now, this is what these mice actually look like. We call normal mice wild type. There's a heterozygous, so these are equivalent to the carriers or the parents who might be in the audience who carry a single wild type and single mutant gene. And then this is the mice that we have Fanconi anemia that we've called Fankel-Tat-Del based on this name of this mutation. As you can see, these mice are a bit smaller than their normal equivalents, and this persists for their early lifetime, as seen as what is seen in humans. They have these defects in skeletal development and eye vision and hearing. So, for example, these mice have deformed eyes or shrunken facial features. They have reduced fertility, which is another characteristic of Fanconi anemia. And importantly, they have fewer bone marrow stem cells, and these are measured just at 10 weeks of age. And we're waiting till we have some older mice. We expect that bone marrow failure incidence will go up as they age. And when we culture the cells in vitro, we can see that they have loss of FANCD2 monoubiquination, which I haven't talked about today, but hopefully everyone who understands Fanconi anemia appreciates that this is a characteristic of most Fanconi anemia cells. They don't have FANCD2 ubiquitination, and they're highly sensitive to DNA damaging agents when we culture them in the presence of those agents. So we then made a myeloid blood cell line that she could use to test different editing approaches. Again, Lou and Astrid worked for almost a year on these experiments. And remarkably, using this optimal approach that was developed for the GFP to BFB conversion assay experiments, she was able to obtain over 35% editing of the Fankel mutation back to the wild type sequence. And we can actually kind of mimic what happens with that selective pressure by exposing the cells to DNA damaging agents. Just for 5 days, we can kill off the unedited cells, and we're left just with cells where the correction has occurred. We get this up to 70%, but actually if we take individual clones of these cells, what we find is that many of them are actually heterozygous, which means they've restored one allele or one of the genes, but not the other one. And that's all that we need. We only need to restore one gene. So really what we're actually seeing here is a functional correction of at least one Fankel gene in probably all of the cells in our population. And this result's really only come in the past month, and quite frankly, we're astonished by the level of success that we achieved. And I hope that we can prove beyond doubt with a few more experiments that prime editing is remarkably efficient at fixing at least this mutation in hematopoietic stem cells. And importantly, the cells are no longer sensitive to DNA damaging agents as well. Okay, I'm a little bit over time, so I just want to finish with what are the steps that are still required before we can transfer this technology to humans. So primary editing is a pretty new technology. So obviously there's a lot of people around the world working on it, and a lot of things are going to be discovered. But really for our project, we need to just demonstrate that we can actually use this technology to, the cells that we've corrected can restore normal blood production after editing in our mice. And Astrid Glasser is a postdoc in the lab, actually funded by MRV from January to perform these experiments. We're also looking, Lorna McLeeman is a PhD student in the lab, also supported by MRV, who'll be returning from maternity leave in March to demonstrate the applicability of this technology to human hematopoietic stem cells. We also need to show that prime editing in bone marrow stem cells is safe, and this is work that's being performed by a whole series of collaborators here in Australia as well as internationally. And of course, in the very near future, we hope to, well, we're already working with clinical partners and hope to work with some industry partners towards a world first clinical trial, maybe even here in Melbourne. So in summary, gene therapy and gene editing is going to revolutionise the treatment of bone marrow failure. Initially in inherited versions like Fanconi anemia, but I think it's going to have applications to other patients as well. As Amelia said, part of the what Fanconi Anemia Support Australasia is also trying to do is make sure that we know about all of the Fanconi anemia patients in Australia, and perhaps ask them to enrol in with a potential future participation in these kinds of at least research projects and maybe trials down the line. So you can contact either through the website, or you can also email me directly. Thanks very much.
Thanks, as always, Andrew. Fascinating research and beautifully presented. And thank you for taking us through all of that excellent work. And we're delighted at MRV to be able to support part of that mammoth effort. And I can pretty much guarantee that the first Australian trial will be in Melbourne. Not proof. Watch out for an invitation coming into your mailbox soon for an upcoming meeting regarding that. Cool. So we're delighted. Thank you again, Andrew, with that marvelous presentation, and for being so generous with your time last week and this week as well. And I know that the research that you have presented a part of tonight, because it's such a mammoth effort and a big team, both at St. Vincent's Institute and internationally, that you'll have plenty more to share with us in the future. And it's inspiring to all of the clinicians, all of the scientists, but particularly to patients and their families, that this sort of research is ongoing. So lots of things coming through in the Q&A chat. Please keep them coming, and we will reconvene the presenters for the Q&A at the end. So please stick with us. Let's move on in our agenda. And as I said right at the beginning, we have a stellar lineup, literally and figuratively, tonight. And you've already heard from 2 stars, and you're about to hear from another one. So it's really an incredible honor and a great pleasure to introduce Professor Stella Davies. Stella is the director of the Division of Bone Marrow Transplantation, and being a bone marrow transplanter is a special person in my eyes, because us transplanters, we need to stick together. That clinical division is also very importantly includes immune deficiency at the Cincinnati Children's Hospital, and these things are closely related: inherited immune deficiencies, bone marrow transplantation, and bone marrow failure. And really reflecting her leadership role in hematology and bone marrow transplantation, she's the incoming president of the American Society for Transplantation and Cellular Therapy, a society which I'm proud to be a member of, and is really the leading societal group for transplantation and all things related. Her research is focused on bone marrow transplantation, unsurprisingly, identifying ways of limiting and mitigating complications from that, and selecting and identifying patients who may benefit from transplantation. And Stella's going to present for us tonight updates in Fanconi anemia specifically. So welcome, Stella. Thank you for your time. We really appreciate it. And the floor is yours.
Thank you, David. And I'm delighted to be here today. And I have to tell you, it's the middle of the night and my cats are absolutely thrilled that I'm up at this time. So we may see some furry friends as we go along. So I'm just going to share my screen. And I was asked to talk about new developments and things that are new and different in Fanconi anemia. And I wanted to talk about two quite different things, but both of which I think are very important and important for our communities to know about and think about. So I'm going to start first by talking about something that has been new to us in the last, really the last 4 years, which is the identification of a completely new form of neuroinflammation, inflammation of the brain in persons with Fanconi anemia. And this is something we had not known about previously, and I think is important for us all to be aware of. So every story starts with a patient, and this story starts with a 23-year-old young woman who had Fanconi L mutation, and I've known this person since she was 6 years old. She had a successful unrelated donor transplant for marrow failure at the age of 10, really did very well during transplant. And she presented with an acute onset, sudden onset, but transient, lasted only about 20 minutes, left-sided visual field defect. So she just couldn't see to the left for about 20 minutes, really not very long. And when we talked about it, she reported that she'd also had an episode where she went numb down her right arm, her hip and her tongue, and her speech was a bit slurred for about 20 minutes, 2 years previously. And again, I want to emphasize that this was a fairly minor kind of symptom. Because of these symptoms, we did an MRI. And this is an MRI of the head. And you can, if you imagine the head like a boiled egg, this is like a slice through, like that way. So these are the young lady's eyes here. This is the back of the head. And all of this is kind of normal, but what we're looking at is this area here in the red circle. And you can see it's a bit dark in the middle and it's kind of white around the edges. And this is a different slice, and the imaging is just set a little differently, so you can see better this white rim, which is called an enhancing rim, and the darkness in the middle, which generally means tissue that has maybe died, tissue that is necrotic. So this is quite a big thing. And you can see around here, it's a little bit dark compared to what you're seeing over here. So this is swelling or edema. And the whole secret of looking at X-rays or imaging is to look for symmetry. And you can see here, this is very asymmetrical. So that's your clue that this is normal. If you look elsewhere, the different sides match. So this lesion has a typical radiological appearance of a brain tumor. And this is a very important fact. Any reasonable radiologist seeing this for the first time would think that this is a brain tumor. There are some additional extra clues. This isn't the only lesion that is in the brain. And remember, we're talking about looking at symmetry. And this is a slice higher up through our boiled egg, through her head. And you can see here the asymmetrical lesion down there. But there are other little dots here, the lesions here. So there's something a little unusual going on. And if you look at the back of the brain, the cerebellum, so this is the area around here that controls balance, you can see here there are a lot of little dots. And this is a very striking finding and an unusual finding. And the nurses in our division call this brain measles, because it does look like brain measles. So something else is going on. About the same time that this case turned up, we became aware of a second case, patient number 2, and these were really about 3 weeks apart, which was quite surprising and probably just coincidence. This was a 20-year-old young man, had abnormalities in Fanconi J. He had not required transplant. His counts have been quite good. And he fell after a collision during a basketball game. He had weakness, a left hemiplegia, like somebody with almost with a stroke, weakness of the left arm and the left leg, and numbness for about 30 minutes. And again, it resolved spontaneously. And again, and you know how you go back after something like this has happened. In retrospect, he'd been dragging his left foot when he walked for about 2 months. So here is his MRI of his head. And again, you can see a lesion in about a very similar place that looks quite similar to the first one I showed you. It's white around the edges. It's quite big. You can see the asymmetry. You can tell where it is, the swelling around it. And again, a lot of inflammation, dark in the middle, probably some dead tissue. And again, this would be normally something people would think is, oh, that's a brain tumor. But then if you look around elsewhere, just as we saw in the first case, there are a lot of little dots, other dots. These are not, it's not an only lesion. And if we look at the cerebellum at the back of the brain, we've got our brain measles. Here are a lot of little dots that are there. So what happened with both these patients is that they had brain biopsies, which is a bit of a daunting thing sometimes, but was really essential because both lesions were thought to be tumors, but they were not. They showed inflammation, but no signs of any cancers. And since those patients were identified, we have gone on to find additional other patients with neuroinflammation. I'm showing you here a summary of the first 6 patients, but I've now seen 13 patients with this. I do want to emphasize that sounds like quite a lot, but we looked hard and we looked through a lot of people. So most people do not get this. But if you do get this, it is important. It's important that it is recognized as not a brain tumor. And it's because the wrong treatments would then be given, and treatments that would be quite harmful to somebody with Fanconi anemia. And both the first 2 patients I showed you were treated with steroids. The inflammation calmed down, and while they are continuing to be followed, the lesions have been very slow in causing problems. So if we look at these first 6 patients, there are some important things that we can learn. 3 of them have had the big dominant mass, the thing that looks like a brain tumor, and 3 of them have not. So 3 of them just have the little lesions scattered elsewhere. The most characteristic finding are these little cerebellar spots, which are a good clue, and calcification within the brain, little areas of calcium that look bright on imaging. And calcification in the brain tell you that these are cells that were damaged some time ago. If you look at the age of these patients, in general, they are young adults. These are not little children. It's happening a little later. It affects male and female people equally. And it goes across the gamut of our Fanconi genes. It's not one particular type of gene. And now, as we watch over time, we see these abnormalities going on for a long time. It's also not a transplant effect. A lot of people have had a transplant just because most people with FA do, but not all. So I think it's a Fanconi thing and not a consequence of the late transplant. And I think one of the things that we've wondered about this is, why are we seeing this now? Why didn't we see this years ago? And I think the answer to that is that now, happily, transplant is working better. We're seeing many more people with FA live into adulthood, and this is generally a thing that we've seen in older people. And we are now having the opportunity to see it. And I do want to emphasize that most people don't get this. This is not inevitable, but I think this is something that it's very important that the community are aware of, know to look for, and know how to respond when it's identified. One of the things that we're working on now is to really understand what the cause of it is and whether things can be reversed or changed. And we have tested a number of anti-inflammatory strategies, the majority of which don't seem to be helpful. In mouse models, there are some parallels, with this neuroinflammatory phenomenon that can respond to alterations in nutritional strategies, and this is the next thing that we're currently investigating. So in this, to summarize this part of what I wanted to talk about, this is a chronic syndrome, and there is a waxing and waning neuroinflammation. These bright lesions, as we follow the children over time, the young adults will come and go, and the acute exacerbations, when we get these big lesions, can be controlled with steroids, which reduce the swelling around them. But that probably doesn't change the underlying process. I mentioned we've seen 13 cases in the United States, and our friends in Germany are probably seeing a similar number. So this is not just a US thing only either. And the awareness of the disorder is important because it must not be mistaken for an aggressive cancer and treated with radiation therapy. So we are testing our anti-inflammatories and modified eating patterns to see if this can alter the course of disease. So I mention here modified eating patterns, and this takes me on to the next topic that I want to talk about. And this is a topic that within our group, the leader is Dr. Lindsay Romick, who is the expert in metabolomics and the director of our Nuclear Magnetic Resonance Center. And Lindsay's focus has been on an issue that I think we've overlooked a little as a research community, which is the nutrition and body size and habitus, how heavy or light you are in people with Fanconi anemia. So this work was really initiated when our very own favorite Australian, Dr. Adam Nelson, was with us in Cincinnati. And Adam was looking at the distribution of body weight in people with Fanconi anemia. And these are data on 122 people with Fanconi anemia, looking at their body mass index. And you know, when we talk about how heavy people are, we talk about body mass index in adults as a measure of whether you're obese or not. In children and young adults, you can't do that because little children are naturally skinny, and you know, just your weight doesn't tell the story. So we use centiles. We measure how close to the average you are. So if you look at a big group of people, you should see a big curve that looks like this. So most people are average. So most people should be in the middle. And here you can see that when people with Fanconi anemia, there's a whole lot of people down here. This is below the first centile. So this tells you that in 100 people, only one person should be down here. You can see here there's a whole lot of people there. This is the 10th centile. This is the 10th to 20th centile. So just as we're perhaps more accustomed to thinking, there's a lot of people with Fanconi anemia who are on the skinny side. But if you look over here, there are quite a few who are at the top end as well. So it's split two ways, and there are many, many fewer than one would expect right here in the middle. And I think we talk a lot about people with Fanconi anemia being on the thinner side, but not much about people being on the heavier side. And both of these things carry their own problems. And this is a little surprising because these people all have the same disorder. And, we looked at the simple things. Is this just whether you're Fanconi A or Fanconi L, something like that? And that's not the case. So what would be going on here? So most people with FA are smaller than expected, except for those who are larger than expected. And what we've found over the years, and those of you who are parents here with children who are skinny, gaining weight, it can be challenging or almost impossible in those who are small and thin. And that's a really upsetting thing to us as parents, right? Because, we feel like when we feed children, we're giving them our love, and it's not working. Similarly, those who have a higher than usual body mass often have issues like insulin intolerance or frank diabetes. So that is not a thing without problems either. So one of the first things Lindsay did to look at this was to look at the distribution of fat. And she found that the distribution of fat in children's bodies, and we have a special machine for doing this, is abnormal in everybody with Fanconi anemia, even those smaller number of people whose body weight falls in the middle. Those kids look pretty normal from the outside. If you examine where their fat is, the fat is in different places and in unusual places to have fat in children. A lot of the fat is visceral. It's in the abdomen, and it's around your liver and the organs within the center of your body. There are abnormal fat pads around the elbows in different places. And visceral fat is an area of where you put fat in the general population, where there's been a chance to study more people. This can cause problems with later heart disease, things like that. The other thing of note is that all of the people with Fanconi anemia that were looked at had much reduced amount of muscle, something called sarcopenia. So if your children are on the skinnier side, you can kind of see that because they have little thin legs and arms. But in the children who are in the normal or the higher range, even those folks who look a little more muscular from the exterior don't have a great deal of muscle. And we've identified some people who have about over 40% body fat, which is quite unusual. So it's evident that there are some very significant metabolic abnormalities happening. So to examine this, Lindsay designed a clinical research study involving normal controls and people with Fanconi anemia to look at these things. And she performed baseline urine collection, blood collection, measuring grip strength, muscular strength, measuring body fat with an in-body analyzer, and performing indirect calorimetry, figuring out how the body uses metabolites. She would give, patients would be fasting, they would then drink a concentrated sugar water, and she would measure the utilization of that glucose, and she would measure with blood and urine what was going on at fixed time points. And the concentrated sugar water contained a labeled glucose, labeled with a heavy isotope that she can trace through metabolism and see where the sugars are being used. So here are some of the key differences in the basic body measurements in the people with Fanconi anemia. You can see here the skeletal muscle index. Everybody with Fanconi anemia had reduced skeletal muscle and reduced skeletal muscle mass. This is a measure of impedance in the trunk region. This measures the deposition of fat here, and you can see that that has increased in Fanconi anemia. The median muscle circumference was reduced, and as you can imagine, grip strength was reduced. So this is showing these features that we talked about of increased visceral fat and sarcopenia, a decrease in the amount of muscle that's present. The indirect calorimetry is one of the kids' favorite experiments, because it's like being an astronaut, but all you actually do is lie under a cover like this, and your breath and your exhalation of carbon dioxide is measured to determine how you are using the fuel, and that's the sugary drink that had been being given. So this is what indirect calorimetry looks like in normal people. So this is the control, and this is measuring your metabolism. It's clicking along, and here the top line is what's expected, and here's the bottom line is what was actually seen. And this is a normal person, and you can see that their base metabolic rate, so how much calories you're using just to live before you work out or do anything, was about 1400, and the respiratory quotient, a measure of how efficient this is, was 0.82. And when this normal person drinks glucose, the things don't change much. Your body is quite good at quickly using that glucose and distributing it. When your body gets glucose, it has to choose, do I need to use that as fuel, or am I going to store it as fat? And that's being made efficiently. This went up a little bit, didn't really change. If we look at somebody with Fanconi anemia, you see something very different. So let's start over here. Here is our expected rate, and here is what was actually seen. So really remarkably lower, to the extent that our dietitian who was working on the study kept telling us the machine doesn't work, the machine doesn't work, and no, it wasn't the machine. So here is the calories being used, quite a bit less than expected at baseline, but here's the response to drinking sugar. The body basically started to hibernate. It had seen one sugary drink and it dropped the basal rate down. And I don't know if any of you had the experience of when your children eat carbs and they want to go to sleep. This is basically what you're seeing here, that the body is switching off and not using those carbs in the normal way. So this showed a very profound difference in how people with Fanconi anemia choose to, and their body chooses, not their mind chooses, to use food, and particularly carbs in this case, when it's put into the body. And basically the body responded a little as though it was going into hibernation or a response to starvation. So Lindsay has tracked, using her heavy isotope, that this glucose is tracked, how it's being used and how it's being metabolized throughout the body. And the choices the body makes in utilization are very different. So how do our bodies choose what to do with nutrition? So these things are generally controlled by a series of different hormones that tell your body what to do. And we're familiar with insulin that controls our use of glucose. And this is abnormal in response to a glucose load in people with Fanconi anemia. You can see that here. This is the people with FA. These are the controls. Cortisol, the key hormone that gives us our energy and gives us our circadian rhythm, is managed differently. There are other hormones like GIP that control how we empty our stomachs, how food progresses throughout our body. Again, you can see that this is very different and abnormal. And these are a series of such proteins, all of them are abnormal. So this hormonal control is what's telling the body to choose and behave a little as though it's starving when it's not. Ghrelin, a very key hormone that is known as the hunger hormone, that tells you when you need to eat, is largely absent in many of these patients, and you can see it here. And I know some of you have kids who are on the skinny side, very small, and you may know that you can jump up and down and do whatever you want to try and get them to eat, and that there is no appetite. And it's really hard to eat when you don't have any appetite. So again, these things are being driven by different hormones. Ghrelin is a hormone that we're particularly interested in, and we're following further, because it does regulate many of these issues and choices that we are looking at. How you regulate your energy expenditure, which clearly is happening wrongly, the signals that you get that tell you to eat. This is an important focus of further investigation. So, just to summarize the second part of the talk, people with Fanconi anemia respond differently to the intake of glucose after fasting. And this really is a very key observation to try and take us down the road of where we want to get, which is, how do I feed my child with Fanconi anemia, which is a really, really important issue. The metabolic response in children with Fanconi anemia ultimately leads to either the excessive burning, using up, or abnormal accumulation of fat. So in the smaller children, the fuel is being used straight away. The muscles are small because the body uses burning of muscle as a reserve for times of extreme starvation. Generally in the West, we don't starve, so we don't lose our muscles, but our children with Fanconi anemia are losing, even those who have reasonable amount of fat. We are seeing the accumulation of visceral fat, which is concerning for later metabolic health and prevention of heart disease, things of that kind. And we're seeing the lower muscle mass, and again, this is part of the starvation kind of phenotype, that the body's making the wrong choices because the wrong hormones, the wrong instructions are being given. So where is this going to lead us to? We hope it's going to lead us to interventions in how we feed children with Fanconi anemia, what we feed them with, and it's possible that we could augment some of these incorrect signals, like we're hoping that our focus on ghrelin it's possible that replacing ghrelin would lead to improved use of fat, a more normal body type, as these things are important, particularly when people are very small. That can be something that is difficult to handle as the children grow older, and it's not healthy. These problems lead to other medical issues or challenges during times of severe illness, that there is not as much reserve as you normally want there to be. So we're testing things to make things better. Should we replace ghrelin? Can we alter the content of the diet? Can we add other supplements? It's possible there are other dietary supplements to do this. And we are interested in the timing of eating, whether it benefits it. So we will tell you as soon as we know what's best to do, because I, as a parent, I know this is a very important issue. One last little piece is one of the challenges of understanding nutrition in children is that we can't look directly at their gut, and while mice can be used to model Fanconi anemia, haven't been the best and most accurate model. So we are making organoids, little tiny tiny weeny little pieces of gut that grow in the tissue from people with Fanconi anemia. We can start with small amounts of cells taken from things as simple as a skin biopsy to determine how the disorders of the Fanconi pathway impact the growth of the GI system, its development, which may commonly have abnormalities in Fanconi anemia, and how it behaves. So here we can test things like supplementing peptides like ghrelin to see if it will, it'll be a model to help how things go. And these are some of Lindsay's little intestinal organoids growing in the Petri dish and showing the presence or absence of ghrelin. And you can see here this is GOAT. This is the enzyme that makes ghrelin, and you can see here this is somebody who is normal, has Fanconi system. This is somebody who doesn't. You can see that these things look abnormal, but GOAT is present, the enzyme that makes ghrelin. But here you can see that ghrelin itself is almost completely absent. So this gives us a model to start to work with and study. So I want to say thank you to all the patients with FA, all the families who are on here. These are some of our lovely patients and lovely families, and to my colleagues, Adam Nelson and Lindsay Romick, who we all, one of the joys of Fanconi anemia is to, it's a community and we all work together. So I'm really glad to have the opportunity to speak to you and hopefully be part of your community. Thank you very much.
Thank you very much, Stella. That's fantastic work and incredibly well presented and very well pitched for people who perhaps this isn't their core area of research or knowledge. So thank you, and particularly thank you for joining us with your cats in the small hours of the morning.
And my latte.
Okay, good. Well, very important. So we are going to convene a Q&A session now, but I am mindful that it is the middle of the night for you, Stella. So if you wanted to sign off early, would be very understanding of that. And perhaps maybe we can direct perhaps questions to you initially, if that's okay.
Oh yeah, I'm fine. I'm up now. I'm up now.
Okay,
Yeah.
all right. Well, that's even more generous of you. Thank you very much. So I might just ask some of our panelists to restart their videos so that we can see people. And I do want to introduce Mei Ling, who's just joined us here on the video, who is our telehealth support nurse, does excellent work with a number of families, a large number of families, giving them advice. And I think something that Amelia said during her presentation, which I think is core to Mei Ling's role, is helping patients and their families understand the right questions to ask. And so I think that's something that you hit upon completely independently there. And here's Mei Ling's details if anybody wants to jot them down, or feel free to contact Lou or Jaya at MRV to get those details if you'd like to talk to Mei Ling. So I think if I'm playing the game correctly, we are into our panel discussion and Q&A session. And thanks to everybody who has given up their time to join us today from around the country and indeed around the world. Let's so if I do my chairing job, let's just see if we can direct some questions, and we will start with you, Stella. So we've got a mixture of patients, family members, support crew. We've also got clinicians and some researchers in the audience, and I'm going to ask a question on behalf of one of my hematology colleagues. So the neuroinflammation is, I think, probably news to us all. It's clearly something that you have taken a key interest in. And Patty asks, what do you feel is the best therapeutic pathway? And do the patients, the young people generally, have they shown a pattern of recurrence, or have you been able to prophylax against that inflammation to intervene with the natural history?
So the answer to the last question, have we altered the natural history? I would say no. So in the large majority of patients, and I have one patient who, the other patient is different. The disease is present and it generally continues, and there is some progression. So if we watch the MRI over time, you will see some of these lesions appear and disappear a little. They become brighter, they enhance, and then they go down some. So many of them are silent. They're small and they are silent. But the cumulative effect over time, and I'm talking about, years now, is that there is loss of cognitive function and there's loss of skills. And the interventions we have made is when there is a large lesion that has edema around it, and remember we saw that swelling, that responds to dexamethasone, as you would imagine, and that will go down. And the enhancement, the brightness, the inflammation itself becomes a little more quiescent, but some of those lesions go up and down, and new lesions, new bright enhancing lesions appear over time. So we have looked at, if you in the biopsy, you see some macrophages, not a very dense lymphocytic infiltrate, necrosis in the center. So interventions like anti-T cell therapy, sirolimus, anakinra, tocilizumab, probably aren't making an enormous amount of difference based on what we've been seeing. As I mentioned, Jan Wijmakers in the Netherlands has a mouse model that looks somewhat similar to this. And in those animals have sarcopenia, they have neuroinflammation, they have shortened lifespan, a number of important issues, and altered feeding has completely modified that phenotype. So what Jan does with those patients, those mice, is calorie reduction. Calorie reduction, we think, is not particularly realistic in our patients, but intermittent fasting might be. So that is something we're trialing now as an easy intervention that could be a lifestyle change that one can realistically do for years and forever. But this remains very challenging, and there is a progressive loss of skills and function. It's really slow, but there is over time. When we first identified these cases, and remember I told you that two of the cases were seen within 3 weeks of each other, it felt like an epidemic. We thought, my goodness, this must be everywhere, and we've haven't looked for it, so we haven't seen it, and everybody gets this. That is not the case. So we went back to every MRI we'd done at our center. We MRI'd everybody. Our friends in Minnesota did the same thing. Our neuroradiologist in Cincinnati reviewed everybody's MRIs. So we're talking about over 200. And we did not find anybody who had this that we did not know about. So there was not a whole big pile of asymptomatic people sitting there just waiting for this to be found. So that was a great relief. So I do want to stress this is happening in a minority of people, not everybody and not most people. But we do, as our children reach the age where they can do an MRI without sedation, we do a baseline MRI on everybody just to know where we're starting out.
It's very helpful. Thank you for that answer. And thanks to Paddy for posing the question. Paddy's been quite busy on the chat tonight, so we might just skip over a couple of his and come to some other questions, but maybe circle back to those points. I personally would like to ask Amelia a question because I have a number of patients, as you might guess, with various forms of bone marrow failure syndromes. And one of the things that was very clear from Amelia's presentation and some of the introductory comments from Andrew and backed up by Stella's presentation, is that Fanconi is much more than just the bone marrow. We sometimes forget that because many of the people who are at the pointy end clinically are the bone marrow doctors, but of course it's much more than that. So Amelia, you made towards the end of your presentation, you made a bit of a list of all the specialists who you, almost like the A list and the B list. There's a few sort of coming off the bench a bit later on. But one thing I wanted to ask you, which, and perhaps Mei Ling may want to chip in here as well, is how do you coordinate all of those guys and women? How do you make sure that they're talking the same language and are doing things in a coordinated way? You're clearly a very able woman. Some people are less health literate, and would there be a piece of advice about how they engage their medical attendants to do things in a coordinated fashion?
Thank you for the question, David. I have a couple of thoughts on this. The short answer is yes, it can be really difficult to manage, and especially since moving out of home and starting to learn to manage that on my own, I've really struggled with it a bit. But the main person I have to thank for being able to coordinate my doctors at all is my mum. She used to be a nurse. Hi, Mum. She's probably still on. Hi, Mum, Dad.
Hi, Mum.
And she, when I was going through treatment, she was really helpful because she knew the right questions to ask and which things needed to be written down and which things we didn't need to remember and things like that. And when we started looking at all of these different doctors and all of these different things that we needed to look for, we used the Fanconi Anemia Research Fund's clinical guide partly, and we used the advice of primarily my geneticist who was doing my gene pathway stuff, kind of gave us some guidance into who we should see and got us connected. And then once we kind of had those networks in place, my mum created this amazing Excel template where I have columns of all of my different types of monitoring, like dermatology, ENT, etc., all along. And it has like all of the information about how often I need to see them, and how to get in contact with them, how to make those bookings. And then it has like the months, so I can put all the dates in there. Does take some dedication to get used to and to use, and I've fallen out of the habit a little bit, but it's a really helpful tool, especially as you're trying to work things out and hold everything in your head, basically, so you don't have to hold it in your head. To my understanding and memory, we created that template and have put a copy of it on the FASA website, Fanconi Anemia Support Australasia, which is the charity that I believe a link went up.
Yes,
for it.
Yeah.
So that resource is online, and you can just download it and use it. It's great. Can recommend.
Yeah, it sounds fantastic, and it sounds fantastic, and certainly something that other, if you like, multi-systemic bone marrow failure diagnoses could certainly utilize, because the themes are often very similar.
Absolutely.
Thanks for the answer. We'll go, Amelia, keep going if you've got something else.
Sorry, just one more thing on that in terms of getting doctors to talk to each other. It's also very difficult. But yeah, I just had to give them a list of who I wanted to talk to each other. Didn't make sense for all of them to talk to each other, but you need to constantly be checking that they are sending those emails to the people that you requested that they send them to, which can be a bit exhausting. But my hematologist and geneticist kind of have their fingers in all the pies, which is very helpful as well. So
Yes.
very lucky to have them.
That's great. Thank you. And look, lots and lots of things on the chat congratulating you on your presentation, and the warmth and generosity that you have sharing your story, and many people will be inspired by that and be better educated as a consequence, which is a fantastic outcome. I'm going to move to Andrew now. There's quite a few questions, as you might guess. and some that I might just bundle up. So forgive me if I use the chair's prerogative. Andrew, fantastic presentation, fantastic work. Really can see the near clinical applicability of these approaches. A couple of questions here. One is about, are there particular Fanconi anemia gene abnormalities that are more amenable to this approach? Any that you feel are most amenable, and as a consequence or as a reciprocal, any that you think are just beyond this approach at this time? And I'm talking about gene editing here, but perhaps extended into any form of gene therapy. Ones that are like first cabs off the rank, others that need more work, and those that are just too difficult at this stage.
Sure, yeah. So for prime editing, which is a technology that we've been specifically working on, the application will be primarily in small mutations. So for those people who are not geneticists, there's lots of different types of mutations to DNA, and we can have single letter changes, single letter losses or additions, or we can have large chunks of DNA that get lost. Prime editing will work very well on those very small deletions, very small insertions or changes in the sequence of the DNA. And for Fanconi anemia, we did some work when we started in this area, just looking at the number of patient mutations that could possibly be corrected, and it worked out about 70% would be suitable for prime editing, and another 30% would not be. However, because it's a heterozygous disease, a lot of patients have two different mutations because you inherit one from your mother and one from your father. The likelihood that at least one of them is editable is obviously higher because you have a multiplicative effect. So there's some other technologies, one called base editing, which can potentially add in another 10 or 15%. But for those large insertions and deletions, there are some other newer technologies coming on board now as well. And I think Patty asked a question in the question box about what could be used for those very large deletions, for example, that take out the whole gene. Perhaps those are more suitable to the existing gene therapy technologies with a viral delivery of the intact gene. But even for some genes, that doesn't work because of issues with gene dosage. So you do actually need to edit in the endogenous locus, and there are some interesting novel therapies or novel approaches using Cas9 fused to integrases, which can insert up to 7 to 8 kb of DNA to potentially restore a large missing gene.
Great, thank you. That's great, and I mean, it was so fantastic to have a number of potential platforms that might be applicable to different lesions. Thanks for that answer. Maybe I forgot to go back to Mei Ling, but Mei Ling, in your experience, what do you feel is the most confusing or uncertain thing that patients and families come to you for support to help tease out, and how do you advise them regarding what is and Amelia made the comment when she first got diagnosed, this completely crazy, overwhelming diagnosis, and trying to and really seeking and wishing for more specific support early on in that process and getting it later, and clearly being a great contributor in that regard. Is there a pattern of unanswered questions that you see in the patients coming to you, and not just focusing on Fanconi, but perhaps more broadly in other bone marrow failure syndromes?
Hi, David. Thank you for the great question. And thank you so much, Anaemia, sharing your fantastic, very inspirational story. It's just really heartfelt to hear your story. And then it really reflects our patient and families' journey. Like David said, like you said, and everyone aware, for Fanconi anemia, everyone has got a very unique journey and different experiences, and it's really confronting for patient and family because they don't have anyone to rely on. So it's so important for them to, at the first place, to gather some important information and the support, and that's the role for us to fit in. Yeah. So for us, I'm so sorry. my kids didn't go to bed yet. I'm so sorry about that. So
Mayling's kids and Stella's cats.
Yeah, now is the time. They should go to bed, but they didn't. So I'm sorry about that, everyone. So we do have a lot of patient and family. They do encountering a lot of difficulties in terms of fear, uncertainty, like Amelia was mentioned about, because of lack of information. So it's really hard for them to find out what is next step and what should they go to. And that's why information is quite important. And this is our job, to educate patients and the family. We are here to walk every step with them, and then to guide them how to ask the right question. Like Amelia said, asking right question is very important in order to get more better information, and then for them to make a better informed decision. Yeah, so we always ask them to reflect. Do you really understand what doctor telling you? And do you know what does that mean to you? Are you able to process that? And what can we do for the next step? And what can we help in order to help you to make all of these things easier? And that's what we are doing here. And another thing I would like to mention is we are trying to promote safe management care, safe treatment and care plans. Yeah, like Amelia's mom has done fantastic job there, put up all of the spreadsheet and the information appointment. It is difficult, and it is really hard for bombarded appointments, information overloaded, like all different things all at the same time during the diagnosis time, even during the transplant period. So we are here to help patients to organize make a plan similar to your spreadsheet, like Amelia's spreadsheet, and help them to collect all of the data, and then make sure the next appointment will be on time, and how to coordinate all the appointment all together. So that's the things we are trying to promote here to all of our patient and family. I hope I have answered your question, David.
Of course, Mei-Ling, you always do. Thank you. We're almost near time, but there are some really important questions. I'd really like to thank Chris for posting some of the links and some of the societal information portals. But he also asked a question, but I think Stella's answered it offline, but just not everybody might be able to see that. But just there's a few questions that are linked here. So Stella, if I can direct a sort of a global question to you, trying to pull some of the themes together. I couldn't help feeling as you were presenting the neuroinflammation case series and some of the thoughts behind that. Do you feel that there is any link between, well, do you feel that that is a dysfunction of the endothelium? Is there evidence of endotheliopathy elsewhere? And I couldn't help thinking as a transplanter that we not infrequently see endothelial dysfunction. And if that's not too much of a vanity question from me, do you think there's any link between the vascular abnormality and the metabolic abnormalities that you presented in the two separate parts of your presentation? You are on mute, so thank you.
Yeah, but not enough zooming in my life. That's my problem. So the other area, the only really other area that's been identified as involved in the neuroinflammation is a retinopathy, and there's an exudative retinopathy that can be quite subtle. You need a fluorescein angiogram to see it. I also I'm a great fan of endotheliopathy as a source for many things. There are clearly vascular abnormalities and some in the biopsies of the small number of biopsies that we have. So I think that's a perfectly plausible thing. We've been interested in identifying the triggers within the brain of why does a spot appear at a specific point and not everywhere? This is not a diffuse encephalopathy, it's focal. And we have, in collaboration with Igor Korolnik in Chicago, identified JC virus as being active at those sites. However, whether that's a trigger, a cause, or part of the normal flora of the brain, which is difficult. But we do know that patients with Fanconi anemia handle polyoma viruses differently. The Fanconi anemia intact pathway is needed for virophagy. So, virophagy is a form of selective autophagy, and you need to use it to extrude virus from the cell, and patients with Fanconi anemia don't do that well. And patients with Fanconi anemia will have more BK virus during transplant than regular folk. So those are interesting things. Does the nutritional issues interrelate? I think they have to, David. The metabolic abnormalities are so pervasive. But now, since you just said that, I'm just thinking about the majority of our patients with neuroinflammation are on the thinner end of that spectrum, but so are the majority of people. So I don't know that we've got a representative sample to say whether there's an easy call and it's going to be in people in the thinner end or not. But I totally think they're going to be related in some way because the metabolic abnormalities are so important. Can I just say one thing, David? If people have further questions about the nutrition,
sorry, Stella, keep going. Yes.
I just want to leave my email for people. My email is stella.davies@cchmc.org, and anybody is welcome to contact me. In particular, families if they have questions, or anybody who wants to. We're a community. There's not many of us. We all need to talk to each other. And I put it on the message to Chris. So feel free to share that. And the more we talk, the more we know.
Indeed. And thank you for your generosity again. We have gone 1 minute over time, but I'm actually going to try something slightly different. I'm going to ask each of the panelists to answer in a sentence or two. Imagine that you're meeting a brand newly minted young doctor or young nurse who might be responsible for looking after patients with Fanconi anemia. What would be the one or two things that you would advise them to heighten their awareness of Fanconi anemia? And I'm going to start with you, Amelia.
I guess probably just you are not alone. There's support out there. You there? You frozen.
Okay. No, we can hear you. That's good. Thank you. Stella, one or two sentences for a young clinician coming on into the field that you would highlight to them that they should really know about Fanconi anemia.
It's a lifelong illness, so form a partnership with your patient. You all need to do it together.
Yeah. Andrew, what would you say? What would you be? You've been embedded in the Fanconi community for a long time.
Well, I would say get to be involved in the community. I mean, I've met a lot of clinicians who've come into the community because they had one or two patients and they saw, what a fantastic community it is and what an opportunity there is both for applying new therapies, but also for helping with a lot of support from a strong community. And think about exciting new
And maybe-
technologies when you're treating
Oh,
your
sorry, Andrew, going.
I think about exciting new technologies and new possibilities when you're planning for the next few years of treating your patient, because they're going to be coming pretty quickly.
And Mei-Ling, you're a practicing nurse. What would you tell your young colleagues about bone marrow failure or Fanconi anemia that would set them on the right track when they meet their next patient?
I'm not sure whether or not set them on the right track, but I would say welcome to this exciting world. And then let's work together, provide support and resources to our patient and their family. And it's good to, like Dr. Andrew said, form a very solid, consolidated relationship with them. And we will learn this journey all together. That's what I would say.
Well, thanks to all of our panelists for such open and generous sharing of their views and their knowledge and their research and their insights. And it's been really an absolute honor and privilege to be able to chair such a session. Thanks to MRV for hosting it. But there are many support groups out there. FASA is absolutely fantastic. Get involved with them. You've been shared a number of links and a number of emails. Utilize them freely. We want to hear from people. We want to hear their questions. Remember that this is only the second of our 4. I think we're having a week off next week, because a number of us are actually going to a bone marrow failure conference in Europe, and so we'll be there. And as we have gone a little over time, I'll probably just quickly say thank you again to our sponsors at the top. Thank you to all our panelists, and thank you to Lucy for chairing the organizing committee who has put all of this together, and for the excellent technical and organizational support given by Jaya and Lou, and thanks very much. We're very much indebted to all of them. Apologies if I didn't get to any questions. I think we covered the field pretty well, and it just leaves me to say thanks to everyone for participating. Have a good evening. And for our person who joined us from England, sorry to tell you I'm a New Zealand supporter, so it doesn't worry me that Australia's out of the World Cup T20. New Zealand's playing right now, in fact. Okay, thanks everyone. Have a good night. Join us in a couple of weeks' time.
Thank you all. Bye.